going through arachnoid and dura as you do this. Say, go to a little to the side of that central longitudinal fissure, because otherwise you're getting the foci cerebrae and it's hard. It's going to make it tough. Okay, now you're going to run into problems. What you can do is start to peel it off. Just peel it back.
That's cool.
So now you're exposing that. Okay.
Pull both sides off?
Yeah, you can pull as far as you can, both. You're gonna run into trouble as we get down towards the bottom. Now.
Yeah, that's all I
Okay,
can get.
yeah. The tetorum, remember, is going between cerebellum and cerebrum, so it's going in this plane, which means you have to cut down in to cut it.
Okay.
So what we've just exposed so far, Eric, is we just got part of the dura mater and the arachnoid off.
Okay.
And there was the pituitary. We're gonna lose the pituitary. I hate these big freaking gloves. Yeah, it's not gonna help because my fingers, that's the pituitary.
Okay.
It's sitting right there. The sella turcica would be over it. So all she's done is just expose, what I was saying is, this ridge here, that's the foci cerebrae. Do me a favor, do a cut this way, just right at this point. So cut transverse across it. It's gonna be a little thick and it's gonna be a hard cut. I'm just gonna
This
warn
way?
you. Yep, come closer to me, this way. There you go. Yeah, it's gonna be kinda thick at this point. Keep going. Okay, it should separate. You gotta go down a little bit.
Got
Okay.
it.
Now, look at the tube in there.
Oh, that's cool.
That's this.
Oh.
That's the dural sinus. You see what I'm saying? So the dura mater folds down, makes foci cerebrae, folds back up. There's a piece that stays up here, so you get this open space, the dural sinus
So they go
in there.
this way.
Yeah, so the sinus runs along this ridge.
Okay.
Okay. They call that the superior sagittal sinus because it runs along that sagittal plane. But you can see it at this point really nice. And you
Yeah.
can see how the foci cerebrae then extends below it, this little piece that's extending all the way down. Okay? Now, here comes your hard part. This tetorum goes down this way. So you kind of have to cut into that.
Into this way?
Yeah, into it that way to cut through it. It's going to be thick. Okay, now you want to kind of pull and see if you got it. It should, no, not totally free.
And go this way?
Yeah. Nope, the other way, because just make sure you don't get the cerebellum, which is here.
Oh.
So I'm going to kind of, no, you're going to have to stick your needles in there a little bit more, I think.
Right here?
Yeah, try there. See if we can get it. Nope, it's still attached down below. It's attached in, see it right there? Can you see it in there? Oh, you're cutting it. I can still see it. If you look in here, you'll see a little piece that's kind of in there.
Right here?
It's gonna be right down deep. It's really hard to get. If you cut across this way, take this piece off for now. So we can kind of clear this out a little bit. And I'm gonna kind of, now do you see it in there?
Oh, yeah.
You see what you gotta cut?
Yeah.
That's a tetorum. It's a bit hard, it's gonna be hard. Is it separating?
Yeah, something.
There we go, okay. So now we're gonna kind of peel this out. And you'll see how I'm pulling this crease out? That's the tetorum. So this plate, this ridge,
Yeah.
was sitting in this transverse fissure. That's the tetorum cerebelli. Below that, so above it is the foci cerebrae. Below it, look at this ridge here. That's the foci cerebelli. So the tetorum is in transverse plane. The foci cerebrae and foci cerebelli are in sagittal plane. Okay, now you just peel. I love this because you really see a lot of stuff from this dissection. It has so much good stuff on it. Okay, she's taking off
So
dura
we
and
lose-
araca.
You
Right.
said we lose the pituitary?
Yeah, you lost the pituitary now. There's no way around it. It's now stuck here.
Okay, so that's on the dura mater?
It's just so,
Or, like,
it's
embedded?
so, yeah, it's so wrapped in it that it's almost impossible to get it out. It always comes out. But now you can see all the folds too. If you just even kind of pop this back into regular shape,
Yeah.
you can see where all those folds were.
Mhm.
You can see the tetorum pretty well when you kind of come in here. If you look from that angle, you can see how it was sitting in that fold. I love that you can see the dural sinus too, so you can see that tetorum. You can see how it was wrapping over the brain. Okay, we're gonna leave our dura and our arachnoid, and we're gonna get rid of that. Okay, now, first, probably just find your basic structures. Find me cerebellum.
So this is the cerebellum, right?
Yep.
This whole thing,
Yep, right and left hemisphere,
right and
opposite.
left. Left and
Right.
right.
Yeah, 'cause it's actually facing how I'm facing you.
Yeah.
Okay,
Yep.
so then find me a gyrus.
Right here.
Yep, sulcus, grooves. Okay, now take your probe and insert your probe into a sulcus, and then follow that sulcus.
Like
Yep.
push hard.
Push hard. Yep. Oh, stop. Not too hard.
Mm.
Okay, now, turn it and kind of lift it up in the sulcus. What's that?
Oh, the pia mater.
There's your pia.
Cool.
So you can see the pia. You got to break it to get into the sulcus. That's why I said you got to push
Yeah.
through. You got to push through. Okay, so now let's do lobes. Frontal lobe.
Up here.
Yep. Parietal. Occipital. Come all the way back. Yep. Temporal.
And sides.
Sides. You can see when you kind of tilt, they kind of bulge.
Yeah, right here.
Yep, those are the temporals. Okay. Central sulcus. Be careful. See where the ridge is? Right there. Man, I can't point with these things. This is the central sulcus.
Oh, it goes this way.
Separating the parietal
From the frontal. Yep, on here you can actually see it kind of ridges off. That's it. You can't see the pre and post central gyrus too well on them. They're not too well developed because they're pretty stupid. All right, no, flip us over. Let's do parts of the brain stem. So medulla oblongata. Yep, that first bump. So
Right
this
here.
is spinal cord.
And then this is the medulla.
Medulla oblongata, pons. Yep, midbrain. Okay, cerebral peduncles. I said they're on the anterior midbrain.
So you have to move the lobe.
You don't have to move anything.
Right here.
They're on the midbrain. Yep. See these big white clunky things? Those are the peduncles.
Oh, okay.
Okay.
So anterior midbrain, you're going to see cerebral peduncles. Those are the motor tracks going up. Okay, now flip them back over. Gently separate cerebrum from cerebellum. So just don't bend it in half to break it, but just bend it. Keep going. Okay, now look in there. We lost, this one broke a little bit, but see the 4 bumps? 1,
Oh, okay.
2, 3, 4.
Mhm.
1, 2, 3, 4. Those were the colliculi, posterior midbrain. So inferior, you see it better on this side, superior up here. Sometimes if you get them right, they look like little butts. Okay, now you're going to do a mid-sagittal cut. Don't saw, just cut, separate all the way through, spinal cord, cerebellum, everything.
Like really deep.
Yep, yep, you want to just keep kind of peeling it apart as you do it. You want to go all the way through the cerebellum too, before you separate it. Yep. So now get through. It's like butter, it doesn't take much to
Yeah.
go through it.
It's really easy.
Yep. That's why I don't know why they gave you the steak knives. I usually only give steak knives when we do kidneys, because kidneys are vicious, man. Okay, that side almost looks like the better side right now.
This one?
Yep, to work with. One usually comes out better. Okay, so now, use your probe, find me the cerebrum first.
Right here.
Yep. Corpus callosum.
This.
Yep. Lateral ventricle.
Mhm.
There, that space, below the corpus callosum.
Right here.
Third ventricle.
Mm,
Keep going around this way a little bit
This
more.
way?
Yep, see?
Oh, I see.
See, it's a little split.
Right here?
Yep. You might catch it on this side better. Let's do this one. So
So-
lateral, remember I said, no, go below, corpus.
Oh,
Remember I
this is
said
lateral?
there's a septum pellucinum that separates the two lateral ventricles? That's what you had to break
The
there.
membrane? Oh,
Yep,
okay.
you had to break that to get into that other lateral
Oh, okay.
ventricle. Now, below that, you can see the third better here.
Right here?
There's your third,
Okay.
okay? Now find me the fourth.
A little further down.
Down
Yeah.
here?
You gotta keep going. You gotta go where, see this space that I'm splitting?
Yep.
That's the fourth. So you wanna be down,
Down here?
yep, that little triangle, that's the fourth,
Oh, okay.
okay? So now let's do the connections. What's connecting the lateral to the third? What's the opening? I'm gonna try and find it for you. There we go.
The lateral to the third.
What's the opening between the lateral and the third? What's the prefix for in between?
Inter?
Inter, ventricular,
Ventricular foramen. Okay.
So if you remember, inter is your beginning, and it's between ventricles, so interventricular foramen. So that's the opening between the lateral and the third.
Mhm.
How am I getting to the fourth?
Subarachnoid space?
Nope.
Subdural?
Nope, close. What goes through the midbrain? What's the passage through the midbrain?
Is that the aqueous?
Aqueduct, cerebral aqueduct.
Oh, okay.
Here's my aqueduct. You can see part of it right here.
Mm-hmm.
If I broke through here, you'd see the rest. It's just a tube going through there. So there's the cerebral aqueduct. It's going to go all the way down to the fourth. I'm going to leave the fourth through median and lateral apertures. The median's right here. This would go out to subarachnoid. Lateral will go to subarachnoid. Some will go into the central canal, which is here. Okay. Now find me the thalamus. The thalamus is surrounded by the third ventricle, so I'll give you that hint.
Here?
That's a thalamus. Yep. Hypothalamus.
Underneath.
Yep, go down. Down, that's up. Go down. Further down. Right, see this dark spot right there?
Here?
Yep, this, right there. That's hypothalamus. So here's thalamus, here's hypothalamus. My pituitary was hanging right off here, right off that bottom. That's where we took it off from. Okay, arborvitae.
Wow.
Arborvitae, tree of life. What looks like a
Oh,
tree?
right here, right here.
What looks like foliage. The little gray, see the little gray knobs? They look like
Yeah.
folia. That's on the cerebellum. So the arborvitae's on the cerebellum. The folia are part of the cerebellum.
That's so cool.
It does kind of look like that. Now, your guy is a little lighter than some people's, but you see this network, that's blood vessels, that's choroid plexus. Yours is a little light, usually it's really dark. That's choroid plexus that I'm kind of pulling out here, kind of looks like a, I don't know, a worm. Okay.
So would
So-
you say there was not that much blood going through?
It just maybe, some of them at that time maybe, there wasn't, because some of them are really dark and you can't miss them. They're like these big wads of dark stuff in there. It just depends. Sometimes I don't get a good one and I don't see it really well. So I never ask choroid plexus that way. Okay, so now you're gonna do a coronal cut through the brain.
Which one should I do?
It doesn't matter. And it doesn't,
Right in the middle?
yeah, just go right straight through. Okay. Now you can see cerebral cortex and you can see white matter.
That is really cool.
When you come in. You're also seeing part of the thalamus here,
Mhm.
and you can see how the ventricles look when you come into a coronal cut. So there's your lateral and your third ventricles right above each other. So on this, if I gave you this image, I would just really be looking for white matter or cerebral cortex on that.
And the cerebral cortex is the gray matter.
It's a gray matter on the outside, yep. So that's what you're seeing. That's the image you're seeing when you're looking in the microscope over there. They did just a small section of this in a coronal cut.
Okay.
That's what you're seeing. Okay?
Yep.
That's it.